All right, so this is probably going to be the last video for lab, and this is the last step of our project. Just to recap what we've done so far, first we collected genomic DNA from your unknown algae. That's all the DNA that was in the nucleus and encodes all the genes for that particular algae type. From that then, we used specific primers to target the sequence that's specific for the 18S ribosomal RNA gene. So we weren't looking for the RNA, we were looking for the DNA that codes for the RNA that ultimately becomes a subunit of the ribosome. And that's the 18S subunit of the ribosome. So we PCR'd that fragment and we created billions of copies of the 18S ribosomal RNA gene. Then we took those copies and we sent that out for sequencing by the Sanger sequencing method. And then when we got it back, we get back this file, and I'm going to post this as a Word file that just has all the ACs, Ts, and Gs, and you'll see, when you open that file, you'll see what it looks like. Now, being the last step, we want to take that sequence and we want to compare it to a database of known sequences of known organisms, and hopefully we'll get a match that tells us that our sequence from our unknown algae matches some existing known algae, and then you'll know exactly what algae we had. And then you can go forward with the last project, the last written project, which I'll be describing in an announcement a little bit later this week. So just as a brief overview of what bioinformatics consists of, it's an incredibly huge field, and it's a growing field, and here in the next decade or so, this is probably going to explode as a career path in bioinformatics. We've been able to generate, through these molecular biology techniques that we talked about in lecture and lab, we've generated a huge amount of information. We've sequenced the genomes of not just humans, but dogs and cats and lizards and funguses and maple trees, and you name it. People have sequenced the entire genomes of these organisms, which amounts to almost an unfathomable amount of data. In addition to that, those genomes, then, we also have sequenced specific genes, like the 18S ribosomal RNA gene, from known species. And that's kind of our goal here today, is to try to match our unknown sequence to one of the known sequences that has been entered in this database. So anyway, it's an era of big information. Everybody is collecting all sorts of information from all sorts of different sources. For biology, a lot of that means this sort of molecular biology sequence data. And there's all sorts of really good information in there that nobody has really taken the time to dig out. And so developing tools to interact with this data and to make sense of this incredible quantity of data is going to take time, and it's going to take people. So here's my commercial for future career goals. For anybody who's interested in math or computer science as well as biology, there are going to be some really good paying jobs out there for people who can make sense and pull information out of this incredible quantity of data. So we're going to use this in a very narrow way, and we're only going to look at one sliver of the bioinformatics field, and it has to do with comparing our unknown sequence to known sequences. So the tool we're going to use is a tool that was developed by the National Institutes of Health as a scientific tool for querying different databases about what kind of DNA you might have. It goes beyond that, and I'll talk about that a little bit later. So when you want to find matches to your unknown sequence, there's two general ways you can go about it. You can look at it globally, in other words, try to match the entire stretch of data, or you can look at little slices of that. So in this example, let's say the upper one is the unknown, okay? That's yours. That's your DNA sequence. You don't know what it's from. Maybe you don't even know what this gene does. So you want to search this database for matches to see if you can get a clue as to what this does, or in our case, what organism does it come from? So you can search globally and look for matches that would match the entire sequence that you have, or you can break your sequence down into smaller parts and look for homology in sequences that aren't total. That's called a local search, and as we just said, this is the basic local search method that we're using. So using this method, you can find all sorts of surprising alignments that you wouldn't have expected. So for example, we know that we have algae DNA, but what if when you run that through the database, a hit comes up that matches a fungus? Oh, that's interesting. So somehow that fungus has ribosomes that are somewhat similar to algae ribosomes. I'm not saying that's going to happen. I'm just using that as an example. Let's say you were looking at a gene that actually codes for a protein instead of just coding for ribosomal RNA. You might be putting in a human gene sequence and find a match in Canis familiaris, which is your pet dog. And it may not match the entire gene, but there may be a section of your gene that matches a section of that dog's gene. If you remember back to when we talked about proteins in lectures, proteins have domains. There's membrane spanning domains, there are DNA binding domains, there are receptor domains. So a lot of proteins are mixed and matched with these domains. So you can take part of the sequence of a gene and it gets duplicated and attached somehow to another gene, and you've created a protein that's unique that has the, say, membrane spanning domain of one that's the same as another protein, but the receptor part is the same as a different protein. So you can find these sorts of homologies by doing these local searches rather than global searches. When you do a blast search, it's going to assume that you've entered a random sequence. And that's kind of its a priori assumption, is that you've entered a random sequence and it's going to check that against all of the sequences in the database. It's also going to assume that your sequence has a constant composition. In other words, there aren't going to be tandem repeats or some non-random information in there. Of course, these aren't random sequences and they're not constant composition, but the program has to assume that. From that kind of basis, you can turn up these very surprising evolutionary links. If you find that part of your gene matches part of another gene from another species, that strongly suggests that those two organisms had a common ancestor and have since diverged. So for those of you who are really interested in the math, this program uses something called a Smith-Waterman algorithm. We're not going to get into the details of the math here, but it's going to find the best local alignments, and I'll explain what that is, and it's going to give you a statistical significance as well as a max score. Now with this basic alignment, local alignment search tool, you can compare DNA sequences to other DNA sequences. That's what we're going to do today, and what you're going to do at home. You can also have the program automatically translate your DNA sequence into a protein, into an amino acid sequence, and then compare that to the amino acid sequences of known proteins. If you have an amino acid sequence, you can compare that to other amino acid sequences. If you only have an amino acid sequence, you can have that compared against DNA translation. So the program will take all the DNA sequences in its database, translate them into proteins, and compare protein versus those translations. Or you can do DNA translation versus translation. So all of these different ways of interacting, depending on what kind of starting material you have, you can come out with a lot of different information. And they're always adding tools to this. So there may even be more tools than that now. But again, what we're concerned with today is DNA versus DNA. Very straightforward. So this is kind of how it works. It's going to take your unknown. sequence, and it's going to break it into words. Now, this is a bit dated, and they've changed the program, but the idea is the same. They're going to break your word into 11-letter words, your sequence into 11-letter words. And they'll start with the first 11, then they'll move down a base and do the second 11, move down a base, make this into a word, make this into a word, make this into a word, etc. So that the entire sequence is broken into these 11-letter words. That's called a lookup table, or a LUT for short. Those lookup tables, then, the program is going to take each word and compare it to 11-letter words from the sequences in its database. Remember, this is a humongous database full of thousands, tens of thousands, hundreds of thousands, maybe even millions of different sequences. And it's going to break them all into 11-letter words and compare those 11-letter words to the 11-letter words that were made from your unknown sequence. Now, what you see here on the right is what we call stringency. So if you break it into very small words, say 7-letter words, then you're going to get a ton of hits, and it's not going to be very significant. The matches, you won't have as great a confidence in those matches. It's a little looser. The longer the words are, the more stringent the search is, the more likely that it's going to be a meaningful match. So one of the ways that it scores the matches is to do what's called an identity matrix. And if at a given position in the two sequences, your sequence and your unknown and the known sequence, if your unknown has an A and at the same position the known has an A, you get a score of +1. If it's mismatches, you get a score of -3. So if we look at this sequence down at the bottom here, the C and the C match in position and base, so that's a +1. Then the A's match, and that's a +2. But then you have a GC. Let me clear this out here. The third position, however, you have a G in the unknown and a C in the known. So that's not a match. So you would take a -3. And if you go all the way through this, what you find was there were 19 +1s and 3 -3s. So your max score, or your raw score, would be a 10. That gives you an idea of how many matches to mismatches there are based on the length, the total length of your unknown. Because you're trying to match whole words, you need to have at least 11 letters, 11 bases in a row that all match to get a hit. Anything less than 11 letters in a row being matched, and it won't return that as a hit. So if you look at these two sequences, 98 out of the 162 bases are a match, but there's no single stretch of 11 base pairs that match consecutively. And so this would not return the sequence as a match. You would not get a hit on this. So it has its limitations. I think 98 out of 162 shows a significant amount of homology between these two sequences, and there may be some evolutionary relationship between these two sequences, but because it didn't find that minimum word size of 11 consecutive bases matching, it wouldn't return this as a match. So this is a website. So if you want to pause this and type this into your browser, your URL, the URL box in your browser, that will take you to the website that we use to do these searches, to do the BLAST searches. And it does more than that too. It takes you to the home page for the NIH site. So you'll see it looks something like this. This is a fraction of the page. I just took a little screen grab and cropped it down. A couple things I want to point out here. This is more than just BLAST search. I've highlighted where you want to click to hit the BLAST, but this is where you get access to PubMed, which is a valuable resource. Also, if you look right here, this link takes you to what's called a bookshelf. And that's kind of a library of all sorts of textbooks and scholarly books in the general field of medical science. If you go to there and you type in something like, say, endocytosis, it will return a bunch of citations from books, a lot of them textbooks. If you click on those links, it will take you to that section of the textbook that explains what endocytosis is. This is a tremendous resource when you're trying to write, say, the introduction to a lab report and you need a scholarly source for some background information that may or may not be in your own textbook. So make use of that. You can't quite get the whole textbook for free, but if you were diligent about it, I suppose, and typed in every keyword from, say, a lecture, you could get to textbooks that explain each of those keywords, but it would be very laborious. The next one down, and let me just erase some of this so we can see it better. The next link down is called PubMed Central. And PubMed Central is a subset of all the papers that are, all the citations that are accumulated in PubMed. Again, most of these are going to have to do with medical science, but it does go quite far afield. For our project dealing with algae, that's pretty close to the boundaries of what you might find in this database. This might not be the best database. It probably is not the best database to use for your final written project when you're studying about the algae that you find out your unknown algae is. Again, more about that in an announcement later. So PubMed Central is a subset of all the citations in PubMed, and it's a subset of papers that were funded with NIH funding, National Institutes of Health funding. Because that's government funds, because that's taxpayer money that's funding this research, the law says that all of the resulting scientific papers have to be available to the general public for free in their entirety. So any citation you find in PubMed Central is going to have access to the full paper, the introduction, the abstract, the introduction, the methods, the results, and the conclusions, the entire thing, and the literature cited. So it's a handy place to go, particularly if you're not going through our library's website, because you'll be guaranteed access to the full papers. So that's enough of a commercial for this website and what NIH has to offer here. Let's go to the BLAST website. When you click on the BLAST link, you'll see there's a lot more on the page, but you're going to look for this part of the page that is a nucleotide blast, because that's going to compare, as it says here, nucleotide versus nucleotide. So DNA versus DNA, that's what we're going for here. If you look around the page, you'll see these other BLAST searches are for some of the other types of searches like proteins versus translated DNA, or translated nucleotides versus proteins. But we're concerned with this nucleotide blast. When you click on that link, the top of the page, you'll see this box that says enter accession number or GI or FASTA sequences. What you're going to do is open up that Microsoft Word file that contains the sequence from your unknown algae, highlight the sequence, copy it, then come to this website, click in this box, and paste. Don't worry if it doesn't come out right, if it's misaligned, if there's weird tabs or returns or spaces. The program's going to ignore all of the spaces and tabs and returns and just read the letters. So don't worry about the formatting when it gets pasted into this box. Now, if you scroll down that page, you're going to see a lot of different options. Some of the options will give you a chance to select what species you want to search against. For instance, I think it'll let you search the mouse genome. That's not going to be particularly useful for us because we think we have an algae, not a mouse. So we're going to leave all the defaults the way they are. So you don't have to play with any of those, but you might want to just kind of look them over and see what else is available, the kind of choices that you can make. So once you've pasted your sequence in, scroll down towards the bottom of the page until you see this section. And this is where you're going to make certain choices. what you see up here with these radio buttons that say highly similar sequence or more dissimilar sequence or somewhat similar sequences. That's what we talked about in terms of stringency. The highly similar sequences are going to use the longest words and compare the longest words available. And that's, I think that stands at 36 letter words. The more dissimilar sequences are going to use smaller words, and the somewhat similar sequence is going to use the very shortest words, which I think there's now longer than 11, but it may still be 11. I'm not sure about that. So the way I would go about this is I would start with the highly similar sequences because we really want to know exactly what kind of algae we have. If you do that blast search by clicking blast, it goes to a page that it updates you periodically telling you it's working on it, it's working on it, it's working on it, and then it'll finally give you the hits. If it doesn't come up with anything on a highly similar search, then you go back to this page and choose a more dissimilar search and work your way down. Hopefully we'll get a highly similar sequence though, so you'll only have to go through this once. Once you get through that and it's found your data, it's found some hits. On this search that I did, I don't remember what sequence I entered in, and this is too small for me to read even with my glasses on. It'll return hits. It'll tell you the title that was given the sequence by the investigator that first entered that sequence. So these researchers sequenced a gene from some organism and then entered that sequence in, and this is how they titled their sequence. And you can get some information from that. But over here are your results. These are the important results for your project. Let's see here. We've got max score, total score, query, and then one really important one is the e-value. The max score, if you remember back to that grid when we were adding +1 for matches and -3 for misses, that's what you're going to get from the max score. And then the next really important information is your e-value. And notice that these e-values, I believe that says 8 times 10 to the -116th or 15th. I need to clean these glasses. Either way, that would be a decimal point and then 116 zeros followed by an 8 rather. In my book, I would tend to round that to 0. So these are extremely small numbers for these particular hits. Well, what does that really tell you? What does an e-value tell you? This gives you a pretty good description and some links if you're really interested in the math behind them. If you're really interested in bioinformatics, I recommend this BLAST course that gets you more in some greater details about the inner workings of this program. But to put it as simply as I can, you want an e-value as close to 0 as possible, because what that's telling you is the chances that the hits that you got happened just by random chance. So when we look at the hits that we got on that last page on the previous slide, 8 times 10 to the -116th, the odds of that match happening just by random chance are extraordinarily small. And at some point, the program does finally round to 0. So you may get a good enough hit that your e-value will be 0.0. Or you may get a pretty good hit that's 8 times 10 to the -34th, which is still pretty darn close to 0. If you're getting hits with an e-value that is, say, 1 times 10 to the -3rd or 1, if you get a hit that gives you an e-value of 1, that tells you it was just random chance. You just lucked out. And there you can add no significance to that particular match. If you get a match that's close to 0, that tells you that that's a significant match. That didn't happen by chance, and you can put significance to that. You can count on that being your unknown algae type, for example. So that takes us to the end of our bioinformatics lesson. As I said before, we're both going to Summer and I are going to post the sequences to the lab Blackboard website, and I'll probably try to post a copy of this PowerPoint as well. Look for those in that last module on our great big project, and take that sequence and run it through a blast search and find out what kind of algae you have, because that's what you're going to need to know to do your final written project.